So that brings us to really the new drugs in mantle cell. There are so many things coming along, so many new drugs. We have these contrasts of intensive therapy, which we're moving away from. We'll come to perhaps a little bit at the end of CAR T cells, because I think that's relatively limited in mantle cell so far, although certainly there are studies going on and no doubt some impressive responses. But there are a lot of other drugs that are being explored in mantle cell upfront in combination, in the relapse setting in combination. And we have this interesting scenario where these remain non-curable disorders for the most part at this point. And so as you add, 2 oral drugs as opposed to 1 oral drug, obviously you're going to get more toxicity. Patients being on these drugs long term, the quality of life becomes an issue, and I don't think we do a great job of measuring that. So, and then obviously, as Andre alluded to, the cost of adding in these drugs, and do we use them all together? Do we use them in sequence? So first, John, I think there are a number of different clinical trials using certainly drugs we've talked about, like ibrutinib, acalabrutinib, also venetoclax, which we haven't gotten to, that are being explored in clinical trials. Can you give us a sense of the landscape, not so much yet in clinical practice, but where we're going, and perhaps at upcoming meetings in the next year or so we'll hear more about the role of these new drugs in combination up front?
Right. I think the BTK inhibitors are being looked at in every scenario you can imagine in this setting, in combination with chemoimmunotherapy, in combination with other novel agents. So there's a very interesting combination being explored of using ibrutinib in combination with BCL2 inhibitor, venetoclax, which we haven't even touched on here yet, which we showed in an early trial has activity in mantle cell lymphoma as well. And there's certainly benefit to perhaps that combination or sequencing questions we have to ask. And then, of course, these agents are all being looked at in combination with chemoimmunotherapy. There's an acalabrutinib combination trial with bendamustine rituximab in the frontline setting. That's also actually been looked at in relapse patients. Ibrutinib in combination with R-CHOP and BR as well. And, this is all exciting, but the patients we have today want to know how to do this. And I think really what they're asking commonly is, how do we get away from chemotherapy? And will we be able to do these things as chemo-free regimens in the first line? And it gets to the conundrum about back to transplant and what to do with these patients, because now we have newer agents that could put off a transplant, especially an allogeneic transplant that has significant risk for early death. I think we have to do more trials. We have to do, of course, a better job in this disease, getting more people on a study. The problem is there's just not enough patients to continue to treat off of trials, and because of that, if we do that, we won't learn. I love the combinations. We need to get people on trial, and I hope the audience certainly will understand that's critically important for us advancing this disease, the treatments.
Yeah, so Andre, how do you see this shaking out? Because it seems to me like there's no question, and we'll talk about even some of the newer drugs, but it seems to me there's no question that if you take, say, a bendamustine rituximab backbone and you add to it ibrutinib or venetoclax or acalabrutinib, PFS I would expect to be better. Lots of things improve PFS in mantle cell lymphoma. Overall survival may or may not be different. Toxicity is going to be more, no doubt. How do you think this is all going to shake out? And from the standpoint of long-term, is it going to be better for a patient to get, let's say, and I'm just throwing out a scenario, BR with ibrutinib or BR and then ibrutinib when they progress? And obviously we need the studies, but at the end of the day, trying to figure out, just with PFS as the endpoint, what's the value of these combinations? What are your senses?
It's a really good question, and that's where the real-world data really matter, and have large databases where we look at the impact of the sequence of care. This is true in myeloma, where there's many, more than 10 approved therapies now, but in mantle cell lymphoma, this is going to become also an issue and a question. So I think what the same concept that matters, now we have the new endpoint in mantle cell lymphoma is molecular CR. MRD negativity. So I think if we talk about biological lesion, as I mentioned before, the combinations we should have the same approach. This is simplistic to think that we're going to have enough deeper response frontline just with a combination of Rituxan plus X, Y, and Z biological agent. So it's early, but there's an ongoing study at Anderson looking at ibrutinib Rituxan lead-in as a window, and in combination with venetoclax. And the molecular CR rate is so impressive that the patient who actually have MRD negative, the proposal of the current trial is actually to stop and not do chemotherapy consolidation. And the patient who are not MRD negative actually continue on hyperCVAD, a short course of hyperCVAD. And I think that's where the field, and maybe that could be BR or RBAC for elderly patient. And I think that's using the opportunity of trying to combine these novel agents to try to get a deep response. So that goes around the issue of the dose-intensive, ability to tolerate dose-intensive therapy. And that's what I would do. And then build on a platform of biological agent, try to achieve a molecular CR, and then consolidate with chemotherapy if needed at that time. And comes the question then the maintenance. Will the maintenance matter as much then in that setting? So that adds a layer of complexity. But I want to mention that in a patient from the Lima trial that had an MRD negativity, which you would expect would not necessarily benefit from the maintenance, they still benefited from the maintenance because the MRD reflects the PBL in the blood and in the bone marrow. The bone marrow was more important than the blood, but they still benefited because it shows that there's some potentially residual cells, obviously elsewhere, what you can't measure. So all of these are still open questions. Back to what John said, the clinical trials will be really important, but really the way I see it profiling to answer your question was a combination biological agent, achieve a deep response, and those patients not maybe have chemotherapy and then moving away from chemotherapy, and then the others consolidate with chemotherapy.